Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the second webinar in the Neurophysiology 2023 series. Today's webinar is titled Autonomic Regulation of Metabolic and Cardiovascular Functions in Health and Disease, featuring Dr. Kamal Rahmouni, Professor of Neuroscience and Pharmacology at University of Iowa. Today, he'll present his research on the fundamental processes mediating autonomic nervous system control of energy, homeostasis, and cardiovascular function in health and disease. Before we get started, we would just like to acknowledge our partners for this series, the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. Specifically, we'd like to give our thanks to today's host society, the American Autonomic Society, for partnering with us on Dr. Rahmouni's event. We'd also like to say thank you and give a shout out to all of our series sponsors. None of this would be possible without them. And without any further delay, I'm pleased to welcome Kamal to join me. Okay, please take it away whenever you're ready.
Thank you. Thank you very much, Sydney. I also like to thank the American Physiological Society and the American Autonomic Society for putting this together and for the support and the work that went behind the doors to make this series possible. I'm very happy to share our work that relate to the autonomic regulation of metabolic and cardiovascular functions in health and disease. So as many of you know, there is an ongoing epidemic of obesity that had touched basically almost every country in the world. And this is due to unlimited access to food and an increasingly sedentary lifestyle. And this epidemic of obesity is especially true in the United States, where about 2/3 of the population is either obese or overweight. As you can see in this CDC map depicting the prevalence of obesity by state in the United States, that there is a high prevalence of obesity in certain states, particularly the Southeast, where obesity can be over 50% in some states or 45% and up. And this is a problem because obesity is associated with many life-threatening diseases, such as high blood pressure or hypertension, type 2 diabetes, liver disease, coronary disease, and so on. And this leads to basically an increase in morbidity and mortality, and is expected to reduce the life expectancy of the United States population. And this association between obesity and some of the disease that are associated with this condition are very clear when you look at these maps. Again, CDC maps. An example here is the prevalence of hypertension. It's another condition that's highly prevalent in the United States. About 30% of the population is hypertensive. And you can see that there is a very tight correlation between the prevalence of obesity and the prevalence of hypertension. So again, the highest level of prevalence of hypertension in the same places where the obesity prevalence is high, the Southeast. And actually, obesity is thought to be a major driver of hypertension in both men and women. About 65% of hypertension in women and about 78% of hypertension in men is thought to be caused by obesity, and this is based on large population studies. But this has been confirmed in both clinical studies and animal model studies. For example, there are a lot of studies showing that weight gain leads to an increase in blood pressure. Conversely, weight loss leads to a decrease in blood pressure. And many of you who may have followed the recent weight loss clinical trials, there are so many published recently, showing that certain drugs, particularly those targeting GLP-1 or the double and triple agonists causing significant weight loss, are commonly associated with a decrease in blood pressure and other risk factors and cardiovascular diseases such as heart failure. Again, this cardiovascular disease is a problem in obesity because it's what's killing people. This is a study that looked at the correlation between body mass index and body mass index is what defines obesity, with a BMI over 25 considered as being overweight, BMI over 30 is considered as obese. And normal BMI is between 18 and 25. So in this study, you can see clearly that relative risk of death goes up as body mass index goes up, especially after 25, BMI of 25, and increase exponentially once in obese people. And you can also see that the risk of death for cardiovascular disease is way much higher than the risk of death by any other causes, including cancer. So this highlights really the importance of understanding the causes of cardiovascular disease and cardiovascular risk in obesity. And we believe that this is a consequence of the crosstalk between metabolic and cardiovascular organs. We know that there is a lot of communication going on between these two entities, and also know that the brain and through the autonomic nervous system plays a major role in orchestrating this communication between the metabolic and cardiovascular organs through its projection, again, for the parasympathetic projections, as well as the sympathetic projections, which are much more predominant than the vagal projections. We also know that the brain receives signals from these organs. A typical signal is the adipocyte-derived hormone that signals to the brain the status of fat mass, but this has consequence on cardiovascular organs, as we will see later. Similarly, cardiovascular organs send signals to the brain, and one such signal is the hormone angiotensin II, but which then has consequences on metabolic regulation. And the importance of these sympathetic and parasympathetic control of different organs is highlighted here in when we looked at the ability of stimulating the vagus nerve that selectively innervate the liver versus stimulation of the sympathetic nerve, again, that innervate the liver. You can see that when you stimulate the vagus nerve or the sympathetic nerve, you cause contrasting changes in blood glucose levels. This is the blood glucose that's coming from the liver. Whereas stimulation of the sympathetic nerves leads to an increase in blood glucose, stimulation of the parasympathetic nerve, so the vagus nerve, leads to a decrease in blood glucose. So there is really a tight control of liver function, in this case blood glucose, by the autonomic nervous system. And this has dramatic consequences in obesity-associated liver disease, such as non-alcoholic fatty liver disease, or NFLD, which is a condition that is highly prevalent in obese individuals. And NFLD is a spectrum of liver disorders that include the presence of hepatic steatosis, and excess fat in the liver induces insulin resistance and inflammation. A few years ago, in a collaborative work with Colin Young, we showed that there is an activation of the sympathetic nervous system in obesity. For this study, we used diet-induced obese mice. These are mice that are fed high-fat diet for several weeks, and we compare these to lean mice that were kept on normal chow. And we used a technique that allow us to record directly the activity of the sympathetic nervous system using electrodes. So we can dissect the nerve to go into any tissue, in this case the liver, and put it on these electrodes so that we can capture the activity going through the nerve. We can filter it and amplify it so we can detect the signal. And then we can quantify it using the computer, so we can detect how much spikes are going through the nerve at any given time. And we use this technique extensively to study the regional sympathetic nerve activity in mice. And we showed in this case, when we looked at the hepatic sympathetic nerve activity, that diagnosed obese mice have significantly elevated baseline sympathetic nerve activity compared to the lean controls. And this can be depicted better here in the quantification data, again, showing that diagnosed obese mice have elevated some hepatic sympathetic nerve activity. Does this has any effect or consequence? To do that, we did a selected denervation of the sympathetic nerve activity going to the liver, and we showed that there is a dramatic decrease in the accumulation of fat in the liver, and this is shown using all-redo staining as well as HE staining. You can see that there is less red in the all-redo staining of denervated mice and less white punctate in the denervated liver as well. And this is translate into a reduction in the hepatic triglyceride levels. So this shows that the expansion of adipose tissue leads to activation of the sympathetic nervous system to the liver, which contribute to the hepatic steatosis. And this activation of the sympathetic nervous system is not restricted to the liver. There are plenty of data out there showing that there is activation of the sympathetic nervous system to many other organs. And these including studies in humans. Is a study that was published by a former colleague, Bill Haynes and Marcelo Correa from the University of Iowa, where they used microneurography to measure the sympathetic nerve activity observing the muscle in humans. And they compared the muscle sympathetic nerve activity between normotensive lean subject and normotensive obese subject and a hypertensive obese subject. What you can see is that the obese subject had elevated muscle sympathetic nerve activity compared to the normotensive lean subjects. You can also see that the hypertensive obese subject have a much higher increase in muscle sympathetic nerve activity when compared to the normotensive obese subject. So these indicate there is activation of the sympathetic nervous system in obese patients. And there are also data showing that this is not restricted to the muscle. Here is an example of a study showing elevated renal norepinephrine spillover in the kidneys of obese patients. Renal norepinephrine spillover is another technique to measure sympathetic nerve activity. And this is very useful for organs that are not accessible by microneurography. And what's interesting in this study is show the correlation between body mass index and the renal norepinephrine spillover, suggesting that the increase in adiposity is what is driving this increase in renal sympathetic nerve activity. And we have confirmed this in animal models of obesity, particularly in diet-induced obese mice in a conscious state. We showed some years ago that we can do recording of sympathetic nerve activity in the conscious mice by implementing some modification, including reducing the amount of quick gel that is used to bind the nerve to the electrodes. And there is a paper in the handout that you can look at if you are interested in this technique. And we have published follow-up papers with this technique as well. But what we found when we looked at the conscious renal sympathetic nerve activity, we found that obese mice, diet-induced obese mice, have significantly elevated renal sympathetic nerve activity compared to the lean controls. And as a reminder, these obese mice also have elevated blood pressure, as depicted in this figure here, compared to the lean controls. So together, this data shows that the diet-induced obese mice is a good model of that recapitulate human obesity. So together, this data shows too that expansion of adipose tissue leads to an increase in renal sympathetic nerve activity. And we know from the literature and a lot of work that was done by others that such activation or sympathetic activation to the kidney leads to vasoconstriction, water and sodium retention. So the question then is whether this translates to hypertension in obesity. And there are a number of studies showing that this is actually the case. And this is an example of a study that used radiofrequency catheter to denervate the kidneys to ablate the sympathetic nerves using radiofrequency. And you can see that compared to baseline, after denervation, there is a substantial increase in systolic blood pressure, confirming the importance of the renal nerve for the renal sympathetic activation for obesity-induced hypertension. And additional evidence confirming this comes from studies that we have done recently, where we looked at the function of protein complex called the BBSome, which is composed of 8 BBS proteins, including the BBS1 protein. And this BBSome has been implicated in trafficking of receptors to cilia, but our work, we showed that the BBSome is also important for trafficking of receptors to the plasma membrane. So when we deleted the BBSome, or disrupted the BBSome through deletion of the BBS1 gene in the SF1 neurons specifically, these are neurons that are localized in the ventromedial hypothalamus, we found that it leads to an increase in body weight in mice. When you look at the body weight of these conditioned knockout mice relative to the controls, and this increase in body weight is due to an increase in fat mass, as shown here in the picture. MRI pictures are showing the fat in white in the conditioned knockout mice compared to the controls. There is more white in these animals compared to the linear controls. What was surprising in this study is that we found that these mice, despite being obese, they are protected from the increase in renal sympathetic nerve activity. As you can see here, there's no significant change in baseline renal sympathetic nerve activity in the conditioned knockout mice compared to the controls. And this translates in the absence of change in blood pressure. Again, despite being obese, these mice do not develop an increase in blood pressure as indicated by the mean arterial pressure values during the light phase and the dark phase of these animals. So this shows that although there is a coupling between metabolic and cardiovascular function, there is a possibility to uncouple the neurocircuit that regulate energy, homeostasis, and blood pressure. So these triggered interest in understanding the mechanism, how that explain this uncoupling or mediate this uncoupling of the autonomic control of metabolic and cardiovascular function. So one way we looked at this is we looked at anatomical or cellular basis for this uncoupling of cardiovascular and metabolic function. And for this, we used pseudorabies viruses these viruses have a unique ability to infect a neuron, but then to jump from one neuron to the next to basically trace the whole circuit that is involved in the regulation of a specific tissue. And we used pseudorabies, a PRV virus that expresses a red fluorescent protein, which we injected either in brown adipose tissue or the liver. And simultaneously, we injected a virus, a pseudorabies virus, express a green fluorescent protein, a GFP, into the kidney. So this way, we're able to trace basically the projection coming from a metabolic organ versus a cardiovascular organ in the brain. And here is a snapshot of the data when we looked at the projection from the kidney and the brown adipose tissue. Again, brown adipose tissue is in red, and the kidney projections are in green. And you can see that there is a lot of neurons that are either labeled in green, means they project to the kidney, and other neurons that project to brown adipose tissue. And this is true for different nuclei that we focus here on the nuclei that are known to be involved in autonomic regulations, such as the parasympathetic nucleus, the dorsal-medial hypothalamus, and the lateral hypothalamus, as well as some brainstem nuclei, the locus coeruleus and the NTS, the nucleus tractus solitarius. In the NTS, it's very interesting to see that the locus coeruleus have a lot of overlap between the neurons projecting the brown adipose tissue and the kidney, depicted in yellow here. Whereas in the NTS, there is virtually no overlap. All the neurons that project to the kidney are separate from the neurons that project to brown adipose tissue. So this is very interesting. We also looked at the overlap and distinction or lack of overlap in neurons that project to the kidney versus liver, and the same thing in the PVN, the dorsal-medial hypothalamus, the lateral hypothalamus. We saw a lot of neurons that are either green or red. That means they project to one organ to another. There is some overlap. There are a number of nuclei or neurons that project to both. In the locus coeruleus now, we see more neurons that project to the kidney versus those that project to both the kidney and the liver. Very few actually neurons project only to the liver. Now, in the NTS, nuclear structures, in contrast to what we saw with the brown adipose tissue and kidney projections, here we see some overlap between the those projecting to the liver and kidney, indicated again by the yellow color here. But even here, you can see that even those that are separated, the kidney versus the liver, they are intermingled with each other. They are not in separate space as we saw with the brown adipose tissue and BAT. And we're able to construct maps of the projection to the kidney and brown adipose tissue or kidney and liver, where we rank the level of shared nuclei or shared neurons versus absence of neurons. And we're continuing to build these maps. So this will allow us to see what is coupling metabolic and cardiovascular function and what is contributing to the uncoupling, where we see no colocalization of the two neurocircuits. And another way, we looked at these pathways that are coupling and uncoupling autonomic and cardiovascular neurocircuits, is looking at the molecular mechanisms or the molecules that are mediating the separation and the coupling of energy, homeostasis, and blood pressure. And one pathway we looked at is the hormone leptin that I mentioned earlier. This is a hormone, again, that's made by brown adipose tissue and acts predominantly in the brain to regulate energy homeostasis by promoting energy expenditure and decrease in food intake. When this hormone was discovered, it was thought that it's a hormone that protects against obesity, but it turns out it's quite the other way around. It protects from weight loss, because weight loss leads to a decrease in leptin, which then drive food intake and decrease energy expenditure. It turns out that this hormone action in the brain has many actions, regulate many processes in the body, as predicted from its effect on metabolism. It was found to activate the central nervous system, to brown adipose tissue, white adipose tissue, liver, and pancreas and skeletal muscle. It also activate the parasympathetic nervous system, as the liver as well. What really was game-changing was when the leptin was found to regulate the central nerve activity to cardiovascular organs such as the kidney and the vessel, because this has then consequences on blood pressure regulation through regulation of water and sodium retention, renal release, and vasoconstriction in the case of the vessel. And indeed, action of the brain was found to increase central nerve activity to the kidney, and that leads to an increase in blood pressure. And this has implications in a state of obesity when there is an increase in plasma leptin levels, or a state of leptin resistance that's common in obesity. And this was based on a number of studies, and this is one of the first to report this idea of the existence of leptin resistance, because when they looked at the level of leptin in the serum versus the level of body fat, you can see that there is a nice correlation, nice increase in plasma leptin as body fat goes up. And some of these patients have high levels of leptin, but they remain obese and hyperphagic, indicating that leptin is not working properly to regulate energy homeostasis. Otherwise, these people should lose weight in response to this plasma leptin. So again, this is known as leptin resistance, and this is analogous to insulin resistance in type 2 diabetes. And we showed that this has consequences in obesity, in that what we called selective leptin resistance. And this is due to the fact that although excess of leptin in obesity is not able to regulate energy homeostasis, it's able to increase the renal central nerve activity and cause hypertension. And we showed this in different mouse models, including the diet-induced obese mice that I mentioned earlier, that are obese and hypertensive. And here is a snapshot of the data. So again, we used lean mice that were fed a chow diet, or diet-induced obese mice that were a high-fat diet. And here we looked at the ability of leptin to decrease food intake over a 5-day treatment with different doses of leptin. In the lean mice, you can see that leptin caused a a dose-dependent decrease in food intake. And this decrease was blunted in the diet-induced obese mice, again, consistent with the idea that there is a metabolic leptin resistance. However, when we looked at the ability of leptin to cause renal sympathetic activation, a different picture emerged. We saw that leptin was able to increase renal sympathetic nerve activity in a dose-dependent manner. And this response was similar whether the mice are lean or obese. So there is no difference between the two groups, showing that there is no resistance to the ability of leptin to cause sympathetic activation. And when we looked at the ability of leptin to increase blood pressure with radiotelemetry, we found that leptin caused about 9 millimeter mercury increase in blood pressure, and this response was again similar whether the mice are lean or obese. So again, this confirms or allow us to advance this concept of selective leptin resistance, which we propose as a mechanism for obesity-induced hypertension. And although all these studies were done in mice, some findings emerged in humans that support this concept of selective leptin resistance as well. And here is an example of a study from Murray Asler, where we measured or looked at the correlation between renal norepinephrine spillover. Again, is a technique to look at the renal sympathetic nerve activity. Looked at correlation with arterial leptin concentration, and you can see a nice correlation between renal sympathetic nerve activity and leptin, suggesting that leptin may explain this the increase in renal sympathetic nerve activity in obesity. In a more recent study by another Australian group, they showed that leptin is required for obesity to develop. Leptin is required for hypertension to develop in obesity, and this was based on studies looking at the blood pressure in leptin deficient patients as well as leptin receptor deficient patients. So what we found is that relative to age and BMI matched controls, leptin deficient patients have lower blood pressure, systolic blood pressure compared to the controls. Similarly, the patients that don't have the leptin receptor have lower blood pressure, systolic blood pressure compared to the control that are again age and BMI matched. This shows that basically leptin is required for the increase in blood pressure that is associated with obesity, because absence of leptin receptor does not lead to an increase in blood pressure. So again, these support this idea of selectivity in leptin resistance in obesity, in that expansion of fat leads to an increase in plasma leptin, but action of leptin in the brain is enabled to regulate food intake and body weight. By this action or this resistance leads or promote obesity. On the other hand, leptin is able to increase the renal sympathetic nerve activity, which leads to hypertension. So this concept is very interesting, but we were lacking at that time is the mechanism. What explains this selectivity and leptin resistance in the brain? So to get at this question, we start looking at the signaling pathways that are associated with the leptin receptor. So the leptin receptor is a tyrosine kinase receptor. Its binding of leptin leads to its dimerization and phosphorylation of the receptor at different tyrosine sites. And this leads to activation of different signaling pathways, with STAT3 being a major pathway, as is the transcription factor that's shown that is required for the ability of leptin or the leptin receptor to regulate energy balance. Work by us and others, we showed that ERK pathway is also an important downstream mediator of leptin receptor regulation of energy homeostasis. We also showed, and we and others have shown also, that PI3 kinase is another pathway that's involved to some extent in the regulation of energy homeostasis by leptin receptor. However, our work we found that only the PI3 kinase is required for the leptin receptor to regulate the renal sympathetic nerve activity. This is based on a number of studies, including this one here that was conducted in rats. In this study, we measured sympathetic nerve activity to the kidney and brown adipose tissue simultaneously. So this is this activity was these two nerves were measured at the same time. And then we measured the ability of leptin to increase sympathetic activity to both tissues, and shown here in green, you can see that there's a robust increase in sympathetic activity to both the kidney and brown adipose tissue. When we blocked PI3 kinase with the LOI compound that was injected centrally, we found that it significantly attenuated the ability of leptin to increase renal sympathetic activity, but did not affect the ability of leptin to increase sympathetic activity to brown adipose tissue. Again, showing that PI3 kinase is required specifically for leptin regulation of renal sympathetic activity. And we have extended this work using transgenic mice. We took advantage of the availability of 2 mouse models that have a gain of function or loss of function of PI3 kinase signaling. The first model is was carrying a kinase dead P110 alpha protein, and basically replacing the P110 alpha with a kinase dead protein, which leads to inability of PI3 kinase to signal basically through this pathway. And we call this a loss of function model. A second model we used is a mouse where we deleted the P10, the phosphatase P10 that convert PIP3 to PIP2. In the absence of P10, then PI3 kinase pathway becomes activated. And we deleted P10 specifically in the neurons that express the leptin receptor by crossing P10 flox mice with mice that express the clear recombinase selectively in the leptin receptor neurons. And we call this a gain of function model because PI3 kinase signaling is enhanced in this situation. And here what we found that the ability of leptin to increase renal sympathetic activity was blunted in the loss of function model. Conversely, leptin activation of sympathetic activity was enhanced in the gain of function model. Similar picture we emerged when we looked at the blood pressure response to leptin. Leptin increased blood pressure in wild-type mice, and this response was blunted in the loss of function model, but is enhanced in the gain of function model. And we found that this has consequences on baseline mean arterial pressure. Data is shown here. So compared to the wild-type control mice, the loss of function model has a normal blood pressure, but the gain of function model has enhanced or increased baseline blood pressure. So these highlight the importance of this leptin PI3 kinase signaling for the regulation of blood pressure, but also for mediating the actions of leptin. So basically what I showed you is that PI3 kinase is a critical mediator of leptin and leptin receptor regulation of renal SNA and blood pressure, and mediate the increase in these parameters in response to leptin. And in a follow-up studies, we identified the mTOR-SO1 complex that contains the mTOR protein, the mechanistic target of rapamycin. In conjunction, this is a protein complex that's formed by the mTOR in conjunction with a number of other proteins, including the raptor, which is critical for the function of the mTOR-SO1 complex. And I want to highlight here that this mTOR-SO1 complex activates a number of downstream pathways, including the ribosomal protein S6 through phosphorylation. And we use phospho-S6 as a phosphorylation of this protein as a readout of mTOR-SO1 signaling. So here is one piece of evidence that supports this idea that mTOR-SO1 is downstream to the PI3 kinase pathway. So again, we use the 2 mouse model I mentioned earlier, the gain of function and the loss of function of PI3 kinase signaling. And we compared the phosphorylated level of phospho-S6 by immunostaining between the wild type and the 2 mouse model. And we can see here in these pictures, as well as the quantification data, that the gain of function model have increased levels of active S6 proteins, whereas the loss of function model have a decrease in the phosphorylated level of S6 protein. On the cohort of mice, we injected them with leptin and looked again at the phosphate level of S6, and you can see again in these pictures here, but also in the quantification data, leptin increased the level of phospho S6 in the arcuate nucleus of the hypothalamus, and this response was enhanced in the gain of function model, as you can see here, and is blunted or absent in the loss of function model, as shown here again, the data. So together, this data confirmed or showed that mTORSO1 is downstream to the leptin PI3 kinase axis. We took it one step further. We generated a mouse that where mTORSO1 signaling is disrupted by deleting the key subunit, the raptor subunit I mentioned earlier. The absence of this subunit basically disrupts the complex and making it non-functional. And we achieved a selective deletion of the mTORSO1 signaling in the leptin receptor containing neurons by crossing the mice that again express the clear recombinase specifically in the leptin receptor with mice that express flux alleles of the raptor gene. And here is the data of the effect of ICV leptin on renal sympathetic nerve activity and sympathetic nerve activity to brown adipose tissue in control and conditioned knockout mice. So in wild-type mice, leptin increased both the renal sympathetic nerve activity and the B18 sympathetic nerve activity. However, the renal sympathetic nerve activity was completely absent in the conditioned knockout mice, whereas the brown adipose tissue sympathetic response was intact in the conditioned knockout mice. So basically, we were able to dissociate the regulation of sympathetic nerve activity to a metabolic organ, the brown adipose tissue, from the cardiovascular organ, the renal, the kidney, through disruption of this mTORSO1 complex. And these and many other data that I don't have time to show here today led us to conclude that this mTORSO1 complex in the in neurons is a substrate of selective leptin resistance, in that it's required for the renal sympathetic activation, but not for the regulation of metabolic functions. And this then again has consequences on hypertension. So this is one way we were able basically to separate at the molecular level the regulation of energy homeostasis and blood pressure, and we showed that in work again that I didn't share today, that this activation of the mTORSO1 in obesity has consequences in for blood pressure, but not on energy homeostasis. So I'm going to stop here. I would like to thank the members of my lab that have done most of the work I described today. Don Morgan is a guru of the sympathetic nerve recording. Deng Fuguo is our go-to person in terms of molecular studies. Paul Williams, who did a lot of the work related to neurotracing, and many other members who contribute one way or another, as well as collaborators and former visitors and trainee who contribute one way or another to this work, as well as the funding. Thank you again. I thank the American Physiological Society and the American Autonomic Society for the opportunity to share our work, and we'll be happy to take any questions you may have.
Wonderful presentation, Kamal. Thank you so much for that. Okay, we can jump right into our Q&A session. We have lots of new questions coming in, so keep them coming up, everyone. Our first question today. Free fatty acid receptor 3 has been shown to stimulate sympathetic neuronal norepinephrine release. Do you think obesity stimulates SNA via the elevated circulating lipids from the enhanced sympathetic mediated lipolysis, or via a more direct mechanism?
Yeah, that's a good question. So we know that free fatty acids are important for sympathetic activation. What we don't know is what happened in obesity. So there are a lot of data in conditions where free fatties are not elevated. So what we don't know is whether these, for example, like I showed you, leptin is functioning in normal conditions to regulate metabolism. But what happens is as leptin levels goes up, there is developed resistance to leptin. So we don't know if the same thing happens with free fatty acids, but it's possible that free fatty acids are doing it through both ways, through indirect and direct pathways. But I think more work needs to be to sort this out.
Okay. Fantastic. Thank you. Does liver denervation provide beneficial effects in diabetes patients?
I think those studies are ongoing. I'm not aware of data at this point, but there are companies who are testing these techniques, the renal denervation techniques, to try to figure out that whether selective hepatic denervation would be beneficial. But I don't know if the data is out there yet. I haven't seen anything coming out, but it's something that's being done. Actually, there is both denervation or studies to denervate both the sympathetic nerves as well as the vagus nerve, because it looks like even the vagus nerve, loss of the vagus nerve may be beneficial in obesity. So that's need to be seen.
Okay, perfect. Thanks. Our next question is asking whether sympathetic activity and heart rate variability correlate with hepatic or renal sympathetic activity.
That's a good question. I don't know. We haven't looked at that specifically, whether there is a correlation between heart rate variability, but as probably you may know, heart rate variability is used as an indicator of the state of the sympathetic nerve activity. So, but whether it correlates directly to the renal nerve activity, I'm not aware. Like I said, we all use heart rate variability as indices of sympathetic nerve activity, but whether really, and typically we think about it as cardiac sympathetic nerve activity, but whether that's true for other nerves, I don't know.
Okay. Thank you very much. Let's jump into this next question. Do you know perhaps why liver transplant patients suffer majorly from metabolic disorders post-liver transplant?
That's another good question. I mean, from my perspective, it's probably the loss of the nerves that are not there to do their job. But it could be that it's the adaptation that happened during the transplant. But I don't have the answer to that question, but it's just all speculation at this point.
Yeah, I think that makes sense. But thank you for sharing your thoughts. Does obesity change the number of surface leptin receptors due to leptin resistance? Delta renal SNA in your system is accompanied by changes in renin secretion by JG cells.
Okay,
maybe he meant, does renal SNA in your system?
Yeah, these are two different questions. So the first question, whether the leptin receptor levels change, it looks like yes. We tried to use so many mouse models, but we were not able. And that's due to the fact that we don't have good antibodies for the leptin receptor. We tried to make transgenic mice to tag the leptin receptor that have not been successful. But there are evidence in the literature from other people showing that as obesity develops, there is a decrease in the surface expression of the leptin receptor. And that's where our BBSome studies comes in, because we believe that's probably the mechanism that causes that. And we believe that decrease in leptin receptor surface expression may contribute to leptin resistance, but we're not sure yet because we have no evidence, but that's possible. For the second question, whether the renal sympathetic activation to contribute to renal release by the GG cells, we don't have data ourselves, but there is plenty of data in the literature showing that in obesity, particularly obese mice, diabetes obese mice, they have activated the renin-angiotensin system in the circulation, which indicate that there is indeed an increase in renin release, as well as the production of the angiotensinogen, the precursor. But we haven't done a direct correlation between nerve activity and renin release. There's plenty of data, including from a former Iowa faculty, Dipona, who did a lot of work in that field, showing that gradual stimulation of the renal nerve leads to a gradual increase in renin release by the kidney.
Okay, Very in-depth response. Thank you so much. Hopefully that answered your question. Our next one, thanks you for your talk. 2 questions, so we'll start with the first one. Are you able to compare simultaneously the frequency and intensity of SN activation between renal-cardiohepatic systems in health and obesity?
No, we can't do the cardiac nerve activity. That's not feasible for obvious reason, because the technique we use relies on the integrity of the contact between the electrodes and the nerve, and because the heart is beating so fast in a mouse, especially as soon as you put an electrode in close to the heart, it breaks down. We've done it in the past, but it was not very successful, and so it makes it very difficult to do that. We know data from humans that use norepinephrine spillover, that the sympathetic activity of the heart is decreased, whereas the sympathetic activity of the kidney is increased, as I showed. So that's the as far as I know, there is data with sympathetic activity to the heart. Whether that's the case in the mouse is probably unlikely. Based on other measurements, the sympathetic activity of the heart of dilated obese mice seems to be increased, and that's based on pharmacological tools that we use to study the sympathetic activity of the heart. But no, we haven't done the comparison in the same animal or different animals. We can't measure basically cardiac sympathetic activity to that study.
Okay, great explanation. So part 2 of this question, any future plans to examine these molecular pathways under the effect of ACE inhibitors and or metformin?
We did kind of, not metformin, but we did a little study or some work with the ACE inhibitor, where we showed that the ACE inhibitor inhibition of ACE, and actually even blockade of the AT1 receptor, blunt the ability of leptin to increase sympathetic nerve activity, renal sympathetic activity specifically, in mice and rats. So we injected ACE either systemically or centrally, and then we injected leptin and found leptin cannot increase the renal sympathetic nerve activity, cannot increase blood pressure in that situation. But then follow-up study from one of my colleagues, Jesse Gropi, showed that actually the renin-angiotensin system action in the brain, specifically the AT1 receptors, are required for leptin regulation of sympathetic nerve activity. But we haven't done ACE inhibition and look at obesity-induced hypertension. Those studies were done by other people, and then they show that ACE inhibition actually block or reduce the blood pressure in obese animals.
Okay, very interesting. That's great, thanks. Okay, next question also says that you did a great talk. Thank you so much. Is there any sex differences in how leptin activates mTOR in obesity?
We haven't looked at sex differences in this study. Focus predominantly in male mice and rats, and the reason for that is that female mice are typically not thought to develop hypertension due to sympathetic activation. They are thought to, at least before menopause. We know that women who develop hypertension after menopause, before menopause, specifically, does not involve the sympathetic nerve activation. So there are some studies from other groups showing that leptin act peripherally to increase blood pressure in female obese mice, and for that reason, we stayed kind of focused on the males, because that's where we think the mechanism is more relevant. But I guess if we look at the females, very likely we find some differences.
Okay, yeah, something to look into for sure. Is this regulatory system lost in kidney transplant patients with perturbed innervation?
Good question. We don't know. We haven't. I don't know if of any study that looked at that, but it's very likely it is perturbed until the nerve grows again, because it's a mode of denervation basically. But I'm not aware of any study that looked at, for example, leptin after transplant, kidney transplant, what happened there, or for that matter, even in obese mice, whether transplanting a kidney for a normotensive mouse, for example, would normalize blood pressure in an obese mouse. So my guess is well, because we know the kidney is very important for the development of hypertension. It requires retention of water and sodium for blood pressure to go up, so it's very likely going to have an effect.
Okay, great. Were you able to look at circadian levels of leptin and RSNA and or live-SNA activity and then correlate to MAP levels, MAP levels?
Oh, no, we haven't done such. That's a tough study to do. Even though we can probably do now the renal SNA, because we can do the conscious mice, so we can do a 24-hour recording. But the liver, we cannot do it. We don't do the again, for technical reasons, we cannot do the liver SNA in conscious mice. But we don't have data. I don't know if that's the case. And again, I'm not aware of any data that would correlate the two, because again, some kind of activity typically we measure at a given time point. We can measure probably for a couple of hours using microneurography or the way we do it, but it would be very hard to measure continuously for during 24 hours. Yeah, it's a good question. We could probably look at that using, now that you have the technique in the mice, working to do conscious recording.
Okay, great. Let's see. Thank you for the wonderful presentation. What are the reasons for hypertension in lean humans?
Yeah, in lean humans, it's a different story probably. I mean, there are different many reasons. Sometimes it could be sympathetic activation, even though the humans are lean. It could be reasons that make sympathetic activity goes up. But there are so many other things that can go wrong, like activation of the renin-angiotensin system, an example, if there's excess of renin activity or excess of angiotensin production. There are renal mechanisms. If there is a problem with the kidney, for example, it could be due vascular mechanisms. If there is a vasoconstriction, it could be a cardiac mechanism. It could be baroreflex problem. So there are many mechanisms that can drive hypertension beyond leptin and obesity. So there are many mechanisms. One classic mechanism of hypertension in lean people is excess of aldosterone and mineralocorticoid in general. That's a major mechanism of hypertension. But the essential hypertension is multifactorial. It can be different factors coming together at the same time, even in the absence of obesity. So it's really, it depends. It depends from one person to another.
Of course, yeah, that makes a lot of sense. Can you just explain how you collect blood pressure in your mouse models?
So, I mean, blood pressure, we measure it. All the data show today requires to use the DSI radiofrequency system. For those who are not familiar, these are small units that you implant in a mouse. A catheter will be implanted in the carotid artery. Then the unit will detect the pressure in the carotid artery and send a signal to a pad. They allow us to record blood pressure continuously for several days, day and night, and in freely moving mice. So this is basically the state-of-the-art technique to measure blood pressure, because again, it records the blood pressure in freely moving mice without the confounding effect of stress and other factors like it's done by Tilka or other techniques.
Okay, thank you so much. This question, what do you do with patients with a history of nervous hypertension?
Nervous hypertension. I presume this is people with the history of sympathetic nerve activation. That's what the
Perhaps.
speaker meant here. So typically, I mean, I'm not a physician, so I can't really speculate on that. But depend on the person. For example, a lean person, we can subscribe them beta blockers. They work very well to block the central nervous system, so that can help with the hypertension. There are drugs that target the alpha adrenergic receptors in the brain as well, that can lower some nerve activation directly from the brain. But sometimes it can also work by blocking the renin-angiotensin system, because the ultimate outcome sometimes when there is an increase in some nerve activity is activation of the renin-angiotensin system. So the active or blocking the downstream pathways like the RAS system can be beneficial. It could be promoting water release with diuretics. So it really depends on the situation.
Okay, fantastic. We are almost at the top of the hour. I think we can squeeze one last question in. And just to let everybody know, we will follow up with a Q&A report in a couple of weeks that Kamal will graciously provide answers to any questions we missed. So, okay, very last question. How does physical exercise influence the mechanism described?
Yeah, physical exercise. So it's like many other things that cause weight loss, like diet is the same thing, and you've probably heard about intermittent fasting as a way to lose weight, and it's the physical exercise leads to weight loss. Weight loss leads to improvement in all the parameters discussed today. It leads to a decrease in blood pressure, decrease in sympathetic activity, improvement in insulin sensitivity, and so on. So, but there is an additional effect of exercise that's beyond weight loss, which again improves health beyond the weight loss, which involves some other mechanisms that due to some hormones that are being released from the muscle, for example, that are beneficial, but also by improving insulin sensitivity and so on. So exercise and is basically and if possible, reduction in food intake is the best way to manage hypertension if it can work, or at least obesity hypertension when it's possible. But that's really hard to implement very often, and that's why we kind of go for medications.
Okay. Excellent response. Very clear. Thank you so much, Kamal. Thank you for your time and expertise today. It was such a pleasure to have you join us, and I hope that you enjoyed yourself.
Thank you. Thank you very much. Thank you, Cindy. Thank you, APS, and thank you everybody else who attended.
Thank you. In closing, I hope you enjoy this Inside Scientific webinar. Another quick thank you to our partners for helping to make this series possible, and I hope that you all enjoy the rest of your day.